of medicine. For over 30 years, Professor Murad has been active in both academic medicine and the pharmacological industry. He has been cited for fundamentally advancing the understanding of biochemical mechanisms in numerous cell types and tissues and the roles of cyclic GMP and nitric oxide. In 1998, Professor Ferid is an acting chairman of the Department of Medicine at Stanford University and was awarded a Nobel Prize for Medicine for his discovery of new properties of nitric oxide, a colorless and odorless gas that acts as a messenger to tell blood vessels to relax, widen, and lowering blood pressure. And this has applications ranging from treating heart disease and shock to reducing the possibility of a life-threatening condition in premature babies called pulmonary hypertension. Distinguished guests and ladies and gentlemen, we are now honored to have a special keynote address by Professor Ferid on the application of research with nitric oxide to the development of new medicine. Professor Murad, please.
Thank you for the opportunity to be here and visit with you and tell you about my research interests that deals with communication between cells and how cells talk to each other. I became interested in cellular signaling as a medical student and graduate student working in the laboratories of Merle Sutherland and Ted Rall. They were interested in the mechanism of adrenaline and glucagon effects in the liver to break down glycogen to form glucose. And during that research, they discovered the first intracellular second messenger, cyclic AMP, in 1957. I joined their laboratory shortly thereafter and began working with cyclic AMP in several systems with heart, liver, other tissues, and became very, very intrigued at how cells communicated, how cells responded to hormones to produce various biological effects. Sutherland received the Nobel Prize in 1971 for his discovery of the first intracellular second messenger, cyclic AMP. The first notion with scientific data to demonstrate that cells communicate with each other probably came with Pavlov 100 years ago. Pavlov was a Russian physician and physiologist working in St. Petersburg, Russia. He had a patient with a gunshot wound to his abdomen. This patient developed a fistula between the stomach and the exterior. And he noticed that when this patient would see or smell food, he would increase his gastric secretion. He then developed some experiments with dogs where he created gastric fistulas to the exterior, showed them food, let them smell food, their gastric secretions increased. But as you recall from your psychology classes, he conditioned these animals, and when he showed them food, he would also ring a bell. And after a period of time, he didn't have to show them food anymore. All he had to do was ring a bell, and they would increase their gastric secretions. This was the first demonstration that cells talk to each other. The brain, the sensory systems communicated with the gastrointestinal tract to modify its function. Well, Pavlov received the Nobel Prize in 1903. He was the third person in medicine to obtain the Nobel Prize. Today we know that lots of cells communicate through this kind of principle. And the first slide, if I can have it, shows you a cartoon summarizing this concept. This slide represents 3 different populations of cells in the body. Cell type 1, cell type 2, cell type 3. They can be any cells, but let's call this cell a neuronal cell, a brain cell. This cell is going to produce a substance that it releases into the bloodstream, and we call this substance a first messenger, a term coined by Earl Sutherland in the 1960s. Today we call these first messengers all sorts of things. We call them hormones, we call them neurotransmitters, we call them autocoids, we call them paracrine substances, we call them cytokines, we call them growth factors. There are hundreds and hundreds of these first messenger molecules. Some of them have very, very simple structures, such as amino acids. Some of them are peptides, some of them are complicated proteins, some of them are lipids. They can be a variety of materials. Sometimes they're bound in the bloodstream to protect them from degradation. They're only active in their free state when they dissociate from these carrier proteins. And what they do is traverse the bloodstream seeking a target. They want to go influence a cell, and they will identify a cell to influence based on the presence of a protein embedded in the membrane of that cell. This protein is called a receptor. The interaction of these first messenger molecules with the specific receptors, like a key in a lock, is a very specific process. Only the cells that possess the appropriate receptors will respond to the appropriate first messenger, which is a hormone or neurotransmitter, etc. The hormone does not have to enter the cell to influence the biochemical physiological processes inside of that cell. This interaction turns on a biochemical cascade that influences the synthesis or accumulation of an intracellular second messenger. The first such second messenger was cyclic AMP. Today we know there are 10 or 12 or 15 such second messengers. Cyclic GMP, nitric oxide, diacylglycerol, calcium, some peptides, some eicosanoids. So there are a variety of them, but not very many. Not as many intracellular messengers as second messengers. Now, what is unique about nitric oxide as an intracellular second messenger is that it's a free radical with an unshared electron, and therefore reactive. It's a gas. It's lipid soluble. It is not charged with physiologic pH as are other second messengers. So most second messengers stay within the cell in which they're made, unless there's a mechanism to transport them or move them out of the cell. Nitric oxide, on the other hand, because of its lipid solubility, with a partition coefficient between lipids and water about 20:1, can go in and out of cells quite readily. There's nothing there to block it, prevent it from going elsewhere. So what is very interesting and unique about nitric oxide is that not only does it function inside of the cell in which it's made to cause an effect, it can also come out of the cell and influence the biology of yet other cells. It's the only messenger that is both intracellular and extracellular. It can influence adjacent cells. If this is an endothelial cell that makes it, it can influence the underlying smooth muscle cell in that blood vessel wall to increase the synthesis of cyclic GMP and cause relaxation of that smooth muscle. It can also feed back and regulate this neuronal cell in feedback systems. And feedback systems can be positive or negative and are very, very common in biology for homeostasis. Next slide. Now, nitric oxide is a very old molecule. Evidence suggests that this planet was created approximately 4 billion years ago. And about 3 or 3.5 billion years ago, oxygen was present on the Earth. But yet there was probably life on this planet before there was oxygen. In the oceans, there were probably microorganisms. And these microorganisms had to figure out how to feed upon each other. How were they attracted to each other to phagocytize and eat, or to colonize and become multicellular? So there were probably some very simple messengers that were available at that point in time. And I suspect in the deep pits of the ocean, with magma coming out and lots of chemicals and lots of heat, there were toxic materials as well as messengers. And the organisms adapted to this environment and started turning around those toxic materials to use them as signaling mechanisms to communicate with each other. I think that nitric oxide was perhaps one of those messengers, as were other molecules such as calcium and phosphate and hydrogen sulfide, etc. So, go back, please. So while this is probably a very old molecule, it did not attract much attention until about 50 or 60 years ago, when it was apparent that it was the product of the combustion of fossil fuels. Whenever you combust a substance that possesses nitrogen in the presence of oxygen, you generate a family of nitrogen oxides. Nitrogen has has 5 different valence states, so you can have a whole family of these nitrogen oxides. And they come out of cigarette smoke, automobile exhaust, smokestacks. The pollution that we see is because of greenhouse gases and nitrogen oxides. And what these nitrogen oxides will do is interact with the ozone layer in the atmosphere to deplete the ozone, to result in greater ultraviolet penetration and global warming, and the environmentalists, of course, were concerned for the past 50 or 60 years about this. However, not only is nitric oxide a pollutant and a toxic material, it's also the mechanism of action of some very important drugs. In cardiology, we use nitroglycerin, nitroprusside for patients with elevated blood pressure, patients with myocardial infarctions, or patients with angina pectoris. These are old drugs. Nitroglycerin has been used since 1870, and these drugs are prodrugs that are converted in the body to nitric oxide. And I'm also going to tell you that most cell types in the body also produce nitric oxide, so it's a normal endogenous substance. And this is a partial list of some of the biological effects of nitric oxide. The first effects that we discovered about 26 years ago were to relax smooth muscle, airway smooth muscle, gastrointestinal smooth muscle, vascular smooth muscle. Why is this important? Relaxation of blood vessel smooth muscle controls blood flow tissues and organs, and the delivery of nutrients and oxygen also controls the blood pressure, also controls the workload on the left ventricle of the heart. It participates in memory, brain, neurology, with strokes and cardiovascular attacks, emboli and cardiovascular lesions in the brain. It is responsible for hormone production and release, such as with insulin in the pancreas. It participates in smooth muscle motility in the gastrointestinal tract, the motility of the intestine. After cyclic AMP was discovered in 1957, a couple of chemists administered inorganic radioactive phosphate, P32, to rats and isolated the urine and purified from urine 2 major organic phosphates, cyclic AMP and cyclic GMP. This was the first demonstration that cyclic GMP was a natural substance and function. Why was it there? Some of us suspected that it might also be another second messenger similar to cyclic AMP. So this stimulated some laboratories to begin to look for the enzyme that made cyclic GMP. And indeed, there are a family of enzymes. We call these guanylyl cyclases. They have a lot of homology and similarity to the adenylyl cyclases that make cyclic AMP. They convert GTP, a cousin of ATP, to cyclic GMP plus pyrophosphate. The cyclic GMP that is formed can be hydrolyzed and inactivated by a family of phosphodiesterases, about 9 or 10 of these enzymes. Some of them specifically hydrolyze cyclic GMP, some specifically hydrolyze cyclic AMP, and convert them to the corresponding 5' monophosphate. Another laboratory started looking for the enzyme that might be regulated by cyclic GMP, analogous to the cyclic AMP-dependent protein kinase that was regulated by cyclic AMP to regulate enzymes such as phosphorylase and glycogen synthase and some of these. And sure enough, there was a cyclic GMP-dependent protein kinase present in tissues. Well, it turns out that there are about 150 different protein kinases in tissues that are activated by a variety of intracellular messengers. These enzymes will phosphorylate proteins, various proteins, by transferring the terminal gamma phosphate of ATP to the protein substrate. The phosphate goes on to a serine or a threonine or sometimes a tyrosine residue. There are many hormones that influence serine and threonine phosphorylation. There are other hormones that influence tyrosine phosphorylation. And this is basically the mechanism of cell signaling. When we talk about signal transduction, we talk about a ligand or a hormone or an agent which through its receptor will regulate the accumulation of an intracellular second messenger, one of 10 or 15 different ones, which in turn now activates a selective protein kinase to phosphorylate a variety of proteins. When these proteins are phosphorylated, their conformation can change, their shape, their motility, their rigidity. These can participate in the motility of organisms, the presentation of pseudopods, the phagocytosis of organisms by white cells. But some of these proteins are enzymes. And when these enzymes are phosphorylated, their activity can change. It can be increased or it can be decreased. And there's examples of both occurring. So there are lots and lots of proteins in tissue that get phosphorylated. And these proteins play a role in so many different aspects of biology. Cell growth, cell cycle, replication rate, glycogen metabolism, fatty acid metabolism, et cetera, et cetera. Next slide. So it was apparent in the late 1960s, early 1970s, that cyclic GMP was a natural substance. There were some enzymes that participated in its synthesis, its inactivation, and perhaps as a receptor of the cyclic nucleotide to mediate some effect. So this encouraged us to believe that cyclic GMP might be an intracellular second messenger to mediate the effects of various hormones and drugs. And at this point, I became much more interested in the potential and prospects for cyclic GMP than cyclic AMP. So I switched my interest to cyclic GMP, and I set out to answer two questions. I said, how do hormones, neurotransmitters, other ligands, regulate guanylyl cyclase activity to increase the synthesis of cyclic GMP? It was known that a couple of hormones could increase cyclic GMP accumulation in the heart and in blood vessels, acetylcholine, some of the prostaglandins. And I wanted to know, were there other hormones that did this as well? And what was the molecular coupling mechanism when the hormone bound to its receptor to regulate the synthesis of cyclic GMP? And if you understand that molecular sequence, you can presumably intervene with drugs to block that pathway or potentiate that pathway, and therefore regulate the accumulation of cyclic GMP in a variety of endocrine diseases. The other question I wanted to answer, what are some of the biological effects of cyclic GMP? If it's present as a natural product, it's got to be important to do something, but none of us understood what it was doing. We began our work by looking at guanylyl cyclase in some detail. Next slide. And in our very earliest experiments, we were quite surprised defined that the enzyme activity in tissue homogenase was both in the cytosoluble fraction as well as the high-speed particulate organelle or membrane fraction. And the properties of these enzymes were quite different. The reason it was a surprise is because this is in contrast to adenylate cyclase, which is found exclusively in the membrane fraction. The enzyme in both the soluble and the particulate fraction that made cyclic GMP behave very differently from each other. There's some kinetic properties. One showed cooperativity with substrate, one did not. I'm not going to go into those details. But this suggested to me that there might be multiple enzymes to make cyclic GMP. If there are multiple enzymes that made it, perhaps they're regulated independently and differently by different classes of hormones to make different pools of cyclic GMP in the cell to perhaps have different biological effects. And I thought this would be a much more challenging problem than working with cyclic GMP. When one characterizes an enzyme in a crude preparation, there is the potential for artifact. There are other enzyme pathways that compete for the substrate that you introduce in your assays. And there are enzymes that compete for the product that you're measuring. And we knew these crude extracts had nucleotidases and phosphatases and phosphodiesterases. So to be sure that we had different isoforms, we would have to purify these enzymes, clone them, express them, and ultimately we did that. But we realized that was going to be a very long-term project. In fact, it took us many years to do all of that, but we have. And sure enough, there's a very complex family of enzymes of the guanylate cyclases, soluble in particulate isoforms. But before we went on to purify and characterize, we said, let's approach this differently. And let's try to introduce some agents that we knew would inhibit the competing pathways. Nucleotidase inhibitors, phosphatase inhibitors, phosphodiesterase inhibitors in our incubations. So we created cocktails of compounds such as pyrophosphate, fluoride, azide, hydroxylamine, methylxanthines, etc. And quite surprisingly found that some of these compounds would activate the enzyme. Now, a couple of hormones would increase cyclic GMP accumulation in intact tissue, but they would not activate the enzyme in cell-free preparations, homogenized. When we homogenized the tissue, we disrupted the coupling mechanism between the receptor binding to its ligand and the activation of the cyclase. If one of the goals was to figure out the molecular coupling, it was imperative that we have a hormone effect in a cell-free system so we could pull apart the components and reconstitute them to validate the fact that we had the right components. But hormones didn't work. But fortunately, we found that a couple of small molecules could activate the enzyme: sodium azide, hydroxylamine, and sodium nitrite. And we wondered if the activation by these substances could lead us in the right direction to understand how hormones were doing. If we understood how these molecules were activating, maybe we could be clever enough to create and reconstitute a hormone effect in the cell-free system, was the rationale. Well, it turned out that the azide effect was tissue-specific. It would activate some preparations, but not all. It required oxygen. It had a time lag before it increased activity at a maximal rate. It was potentiated by reducing agents, particularly thiols. So we said, my goodness, these substances are converted to something else in our incubation. Let's find out what that common substance is. And we approached it in a couple of different ways. The next slide. We did a very simple experiment, basically a reconstitution experiment. We took a liver supernatant fraction where azide would activate, a heart supernatant fraction where azide had no effect, a cerebral cortex supernatant where azide had no effect, and we mixed these. Because they were soluble fractions, we could mix them together. If we mixed liver with heart, the azide effect was blocked. Heart possessed an inhibitor of the azide activation. This inhibitory substance was heat labile, was non-dialyzable, therefore it was a macromolecule, presumably a protein. We purified it. It turned out to be two proteins, hemoglobin and myoglobin. If we mixed liver with cerebral cortex supernatant, the azide effect was potentiated, it was enhanced. And that's because liver converted azide to this unknown activator, but cerebral cortex could not do that. It had the the guanylate cyclase, but it didn't have the factor required to metabolize and activate the azide. The liver had it. The substance in liver that was required for azide activation was also heat labile, non-dialyzable, presumably a protein. We purified it and turned it into a catalyst. Now this was important because we now had a group of heme-containing proteins that influenced the tissue specificity of the azide effect. And that gave us some very important clues as to what this substance was going to be. And this will be apparent shortly. When we found the effects of azide, hydroxylamine, and nitrite, we put them on intact tissue and found, like some hormones, they would increase cyclic GMP. So they would work both on intact tissue as well as in cell-free preparations or homogenizing. And we put them on liver slices and brain slices. One of the tissues, fortunately, that we were working with at the time was tracheal smooth muscle. I had known as a student that cyclic AMP caused relaxation of smooth muscle. It relaxed airway smooth muscle, GI smooth muscle, vascular smooth muscle from my work and other work of others. I thought that cyclic GMP would antagonize the effects of cyclic AMP. If I were to guess at functions of cyclic GMP, I would say in most systems it should probably antagonize cyclic AMP. Well, that was not the right assumption. When we put azide on tracheal smooth muscle preparations, we increased cyclic GMP levels, but the tissues didn't contract. They relaxed. And it became apparent that there was a very tight correlation and association of cyclic GMP accumulation in smooth muscle with relaxation. Not only airway smooth muscle, but gastrointestinal smooth muscle and vascular smooth muscle. So we recognized that one of the biological functions of cyclic GMP was smooth muscle relaxation. When we found that, we said, what do other smooth muscle relaxants do? Well, we tested nitroglycerin and nitroprusside. Nitroglycerin was first synthesized in 1847 by some Italian chemists. It was first used for angina pectoris in 1870. And for more than 100 years, it was used for chest pain with angina, but no one understood the molecular mechanism of action. Nitroprusside is a more recent drug, and that's used predominantly for lowering very elevated blood pressures in patients with malignant hypertension, or diminishing blood pressure in patients with myocardial infarction to decrease what we call afterload of the left ventricle. So we took these smooth muscle relaxants that had been used clinically for a long time and put them into our incubations. They too activated guanylate cyclase. They increased cyclic GMP levels, and they caused relaxation in all of the smooth muscle. So now our list of compounds that were guanylate cyclase activators was really growing. I'd like to be quite a list. In the next slide. Next slide. We developed a list of compounds that we called nitrovasodilators. Now, this has become a popular term that we coined back in the late 1970s. It's in a lot of your textbooks. You see it in Goodman and Gilman and other books. Basically, nitrovasodilators are nitrogen-containing substances that are prodrugs that are converted to nitric oxide. And they include hydroxylamine, azide, nitrite, hydrazine, nitroglycerin, nitroprusside, nitrosoureas such as oxytocin, nitrosamines, and a lot of others. There have been companies that have been making lots of nitrovasodilators for possible clinical use. Isosorbide nitrate, nicorandil, etc., etc. All of these substances are converted to nitric oxide. And we reasoned that the common intermediate with all of these had to be nitric oxide, because hemoglobin and myoglobin were inhibitors of the guanylate cyclase activation by these compounds. And there was some old literature that we found in the 1940s and 50s demonstrating that nitric oxide had a very high affinity for heme in proteins, a very high affinity. So nitric oxide, we thought, was the common denominator that activated the cyclase. If we oxidized it to the dioxide using a variety of agents, we lost the effect. This was the first demonstration that a free radical could activate an enzyme, and this was unprecedented. Free radicals are supposed to be toxic, they're pollutants, they destroy DNA and RNA and proteins and lipids, and that's supposed to activate enzymes. Well, this was a novel bit of biochemistry that we thought could have application perhaps to other enzymes. That's turned out not to be the case so far. But certainly, a free radical can activate a protein guanylate cyclase. When we generated the nitric oxide chemically in the laboratory in the fume hood with ferrous sulfate and sodium nitrite and sulfuric acid and ventilated gas and incubations, we could activate the guanylate cyclase. It turns out that the concentration of nitric oxide required to activate the enzyme was quite low, particularly if we purified the enzyme to homogeneity to remove transition metals and thiols and other proteins that were scavengers or sinks for the nitric oxide. With homogeneous enzyme, we could activate it with nanomolar 10 less 9 concentrations of nitric oxide. So it was very sensitive. And at saturating concentrations of nitric oxide, we could activate the enzyme 400-fold. So it was a very dramatic effect. Slide. This is what a nitrovasodilator will do in a smooth muscle preparation. This happens to be a radiator segment, which has been pre-contracted with norepinephrine to get a stable contraction. And then we add a relaxant, a nitrovasodilator, in this case nitroprusside, but similar effects with any of the other agents. Within 10 or 20 seconds, cyclic GMP begins to accumulate. It peaks in about a minute or so, returns to basal. This is followed by relaxation of the tissue that persists for a long period of time. This is a very common phenomenon with second messenger systems. You see a spike and a decay. The spike can be in seconds or perhaps a minute or two, and then it returns to basal. But the biologic effect will persist for a long time. Why is that? The messenger is labile. It's destroyed, it's removed. Sometimes it's released from the tissue. But once it activates the protein kinase and then phosphorylates all these protein substrates, they have a much longer half-life. So the biology, biochemistry can persist for minutes and 10, 20, 30 minutes or even longer sometimes while the signal is gone. Very common phenomenon. Next slide. So we recognized in the late 1970s that we had figured out the mechanism of nitroglycerin, this popular cardiovascular drug. By being the prodrug converted to nitric oxide, recycling GMP, and then somehow causing relaxation of smooth muscle. Now, when one finds the effect of an exogenous material in a biological system, you should ask yourself, is this mimicking the endogenous mechanism and pathway? The work with opiates, morphine, heroin, led to the discovery of the peptide enkephalins. And there are many examples of that in the pharmacologic literature. So, if nitric oxide mediates the effect of nitroglycerin, I suggested that perhaps the effects of hormones and other agents on cyclic GMP accumulation in intact tissues might be attributable to altered redox in nitric oxide formation from an endogenous precursor. I suggested that nitric oxide was a natural endogenous substance and a second messenger to mediate hormone effects. Well, that was heresy. People were not so excited about a free radical activating an enzyme, but now for a free radical to be a messenger was not received very well, frankly. A lot of skepticism. It took probably 7 or 8 years before others really got excited about it. I was, of course, concerned and upset about that. because I thought it was real, and we had to go on and show it. We ultimately proved it. But I guess in retrospect, it also, because so many laboratories were skeptical about nitric oxide's role in biology, very few laboratories worked with it, and it permitted us to dominate the field. So for many years, we were the major player. We sort of got almost all the important work done before others came in, as a matter of fact. So that helped me go to Stockholm, I suspect. Next slide. Now, in 1980, Robert Furchgott, a professor at State University of New York in Brooklyn, who was a vascular biologist, found a very important observation. It was known for many years that a variety of drugs and hormones, acetylcholine, histamine, bradykinin, thrombin, ATP, a lot of compounds, could lower blood pressure in animals and man. But if you put these substances on blood vessel segments in the laboratory, they did nothing. They had no effect on relaxation. Well, the reason for that was when you make vascular segments in the laboratory, you destroy the integrity of the endothelial lining. And everybody was thinking that the endothelial lining of blood vessels was just an inert lining, like an inner tube in a tire. It wasn't there to do much. Well, they were wrong. The endothelium was required for the effects of these substances. And what was happening is that these endothelial-dependent vasodilators, acetylcholine, histamine, bradykinin, etc., interacted with the endothelial cell to produce a substance that he called endothelial-derived relaxin factor that had a very short half-life of several seconds, very labile, that then migrated to the smooth muscle to cause relaxation. And when I heard his work, I thought that this factor, EDRF, was going to be a free radical or a reactive species of some kind, and therefore it might be working through cyclo-GMP. And that turned out to be the case. Furchgott was working with rabbit aorta, we were working with rat aorta segments, but similar to the nitroprusside experiment, we pre-contracted this rat aorta segment with norepinephrine, and then at 0 time, we added an endothelial-dependent vasodilator, acetylcholine, we used many of them. Within 10 or 20 seconds, cyclo-GMP increased, peaked, returned to basal, the tissue began to relax, and it persisted. Very simple, similar temporal response to the effects of nitric vasodilators. But this only occurred if the endothelium were present. If you remove the endothelium, cyclo-GMP levels did not increase, you did not make EDRF, and the tissue did not relax. In fact, the basal levels of cyclo-GMP decreased about 50%. The activity of cyclo-G-dependent protein kinase decreased, the phosphorylation of smooth muscle proteins decreased, and also cyclic AMP levels decreased as well. So what this experiment told us is that indeed, as suggested by Furchgott, this substance, EDRF, is talking to the smooth muscle to increase cyclic GMP synthesis to cause relaxation. But there is also a substance talking to adenylate cyclase to make cyclic AMP, which will also cause relaxation, and this substance turns out to be prostacyclin. Well, we know today that the endothelium produces lots of substances that regulate the motility of the smooth muscle, that regulate the aggregation of platelets, the tumbling of white cells along the endothelial lining, etc. So it's a very active cell in the blood vessel. Why is this important? There are lots of diseases that are associated with endothelial dysfunction. Hypertension, diabetes, atherosclerosis, cigarette smoking, obesity. In all of these diseases, you don't make enough nitric oxide or EDRF. And so you've got to counter the system, and this is apparent in just another slide or two. Next slide. So in the early 1980s, we recognized there were two categories of vasodilators. The nitrovasodilators, which produce nitric oxide to increase cyclo-GMP, the endothelium-dependent vasodilators, which produce DDRF to increase cyclic GMP. We then went on to show that the cyclic GMP did indeed activate an endogenous cyclic G-dependent protein kinase, and with P32 incorporation into proteins that we were able to purify and identify, there were many proteins that got phosphorylated that resulted in relaxation of the tissue. Next slide. Now this is all summarized for you in this cartoon. This is to represent an endothelial lining in a blood vessel with the underlying smooth muscle. In red are three categories of vasodilators that increase cyclic GMP synthesis in the smooth muscle compartment. The nitrovasodilators in the bloodstream are spontaneously converted to nitric oxide based on the local oxygen tension, pH, other conditions, and the drug of interest. Or they're enzymatically converted to nitric oxide inside of the cell, as with nitroglycerin. The nitric oxide combines with a heme prosthetic group of the soluble guanylate cyclase, which is a heterodimer of an alpha-beta subunit. The nitric oxide binds to the ferrous iron, disinhibits the enzyme, which is inhibited by the heme prosthetic group, and the enzyme becomes activated to convert GTP to cyclic GMP, which activates the kinase to phosphorylate many proteins, which in turn regulate cytosolic calcium, and calcium is required for the enzyme myosin light chain kinase, another protein kinase that phosphorylates myosin light chain. If you don't have enough calcium, you don't phosphorylate myosin light chain, you accumulate myosin light chain in the dephosphorylated state, and the muscle relaxes. Actin and myosin, these sliding filaments. These filaments are pulled. This is governed by the availability of ATP, the myosin light chain kinase, and calcium, because these enzymes are calcium chemologically dependent. The endothelial-dependent vasodilators do exactly the same thing. However, they require an intact endothelium because the endothelium possesses the receptors for these substances. The smooth muscle itself does not have receptors for these substances. So when these ligands bind to their receptor, they happen to increase calcium concentration, which is required for an enzyme, nitric oxide synthase, that metabolizes arginine to form EDRF, which happens to be nitric oxide. So it works the same way. A Canadian scientist, de Bold, discovered that the cardiac atria possess granules and produce a hormone called atrionatriuretic factor or atriopeptins. Other tissues do the same. So the heart is an endocrine gland, the kidney, the brain, they're producing hormones. The atriopeptins relax blood vessels and they cause natriuresis and diuresis in the kidney. And when we saw this new class of hormones, we wondered if they too work through cyclic GMP. And sure enough, they do. It turns out they activate the particulate isoform of guanylate cyclase, which in fact turns out to be the receptor for the atriopeptins to make cyclic GMP. So these three categories of vasodilators work by increasing cyclic GMP synthesis, but you can relax smooth muscle and blood vessels in other ways. You can increase cyclic AMP levels. You can lower cytosolic calcium with calcium antagonists, and that's why you use them in hypertension. You can use phosphodiesterase inhibitors, such as Viagra, to inhibit the phosphodiesterase to prevent cyclic GMP to accumulate. There are various ways to get there. But this is the basis for that utilizes cyclic GMP synthesis. Next slide. Now, nitric oxide is made by a family of enzymes called nitric oxide synthases. The first one was purified about 1990, and over the course of a couple of years, all three isoforms were purified. So there are three isoforms that are encoded by three different genes that reside on three different chromosomes, but there's a lot of homology between these enzymes. They were initially called nodal NOS, inducible NOS, and endothelial NOS, but they're very ubiquitous. They're found in almost all cell types, and we suggested we change the nomenclature. Interestingly, this isoform, NOS2, is normally not present in tissues. The gene, of course, is there, but the transcript and protein are not there, unless the tissue has seen pro-inflammatory cytokines or some infection, because this gene is regulated by endotoxin, by IL-1, interferon gamma, TNF-alpha. It's also inhibited by other cytokines and steroids. So when you see NOS2 transcript or protein, it's because there's been inflammation, as with Alzheimer's or myocarditis or Parkinson's disease or inflammatory bowel disease or rheumatoid arthritis or whatever. Okay? So it's become a very nice marker of inflammation. This is important, and I'll show you why later. Next slide. All of these enzymes catalyze the oxidation of this terminal guanidinonitrogen of arginine, which is an essential amino acid in our diet. It oxidizes it to a hydroxyarginine, and this is further oxidized to nitric oxide. The other product is citrulline. Now, these enzymes, the nitric oxide synthase, have a very complicated array of cofactors. They are calmodulin dependent. Some are regulated by calcium. They use NADPH. They use FMN, FAD. They have a heme prosthetic group, and they use tetrahydrobiopterin as well as oxygen. Why is that important? I think that's the mechanism of endothelial dysfunction. When you have these diseases that don't permit you to dilate your blood vessels with endothelial-dependent vasodilators, it's because you don't have much nitric oxide synthase activity, because you have diminished levels of these cofactors, or they're oxidized because of oxidant stress in the development of free radicals, with diabetes, with hypertension, atherosclerosis, etc. If the cofactors are rate limiting, if the arginine substrate is rate limiting, or if you're producing an inhibitor, an arginine analog that blocks the reaction, what these enzymes will do, instead of making nitric oxide, they make superoxide anion, which is the worst thing you can do, because that'll suck nitric oxide out of the system, as I will show you in a moment. Next slide. Okay, we've come a long way now. We know that a variety of hormones and ligands interact with their appropriate receptors. They then regulate in a variety of ways these cofactors that I just mentioned that are required for nitric oxide synthase to convert arginine to nitric oxide, to activate guanylate cyclase, to make cyclic GMP, to activate the kinase, etc. We can interrupt this cascade with hormone analogs, receptor antagonists, by making these cofactors rate limiting, by inhibitors of the nitric oxide synthase, some of which are arginine analogs and are in clinical trials. We can scavenge the nitric oxide not only with hemoglobin and myoglobin, but we can scavenge it with small thiols, also in the clinic for septic shock. We can potentiate the effect with phosphodiesterase inhibitors, such as Viagra, by increasing cyclic GMP. So we have a lot of tools now to start chipping away at this information to develop some drugs for some important diseases. Next slide. Now, it turns out that not all of the effects of nitric oxide, if hormones and other agents regulate its production, not all of the effects are mediated through guanylate cyclase and cyclic GMP. Most of the beneficial effects are, but not all of the effects are. Because nitric oxide can do other things. It can be oxidized to nitrite and nitrate. It can form complexes with transition metals and form nitrosyls. It'll inhibit aconitase because of interaction with the iron sulfide center and block the production of ATP in mitochondria. It can form complexes with thiol groups in proteins. About 100 proteins have been found to form nitrosyl thiols. Some of these are receptors for hormones, some of these are transcription factors. They're turning out to be important, and maybe this is another way for nitric oxide to regulate cell signaling. Some of these are involved in apoptosis, those are the caspases. A very interesting pathway, I think, is the interaction of nitric oxide with another free radical, superoxide anion. And this reaction is very, very rapid, almost diffusion limited, and faster than the dismutation of superoxide by superoxide dismutase. So if nitric oxide is in the vicinity of superoxide, it goes this way, and it doesn't go over here to activate the cyclase. There are lots of conditions that increase oxidant stress to generate reactive oxygen species and superoxide anion. Inflammatory diseases, infections, etc. In the past, we associated a lot of the problems of inflammation and oxidant stress to the reactive oxygen species. I think that's been an error. I think the real player is the formation of this new product, peroxynitrite, because this is even more reactive than the nitric oxide or the superoxide anion. And whenever you have inflammation in any tissue, you always have evidence of peroxynitrite formation, because one of the things it will do is nitrate tyrosyl residues on proteins. And a lot of us have been identifying these proteins as inflammatory markers for various diseases as an index then of the oxidant stress in that situation. And this occurs in diabetes and hypertension and lots of other diseases. Next slide. How can we start to use all of this now to develop some important drugs? Well, it turns out that nitric oxide participates in neurotransmission. We know that it's involved in memory. If you knock out NOS1 and NOS3 in mice, they have a memory defect. They don't perform well in the water-based test as they swim across the tub to get to a float. If you make too much nitric oxide by NOS1 with a stroke, you have a bigger infarct. If you inhibit the NOS1, you have a smaller infarct. Nitric oxide participates in aqueous humor and vitreous humor formation. It's toxic on the retinal cells, the neural cells in the retina. If you inhibit nitric oxide formation, you can lower intraocular pressure in glaucoma. We've talked a lot about vasodilatation of blood pressure and blood flow. Pulmonary hypertension is really a fun system. Premature children, premature babies, are born with inadequate lung development. They have a deficiency in surfactant, their lipids, which are responsible for helping dilate airways. But they also have very narrow pulmonary arteries in their lungs. They have pulmonary hypertension. Because of this elevated blood pressure in their lungs, they cannot pump blood through their lungs. Instead, they shunt blood from right to left through their patent ductus arteriosus and their blue babies. If you give them nitric oxide by nasal catheter, you dilate these lung vessels. They now start passing blood through the lung. They don't shunt right to left as much. They become oxygenated and they do much better. So around the world now, particularly in the US and Europe, inhalation of nitric oxide is used very commonly in premature babies with pulmonary hypertension, and many of them have it. It's also being used in children with congenital heart disease that shunt right to left to give the surgeons some more time to work out the anatomy and figure out what sort of reconstructive surgery that will be done. It's been less successful in adults with pulmonary hypertension, and that's because pulmonary hypertension in adults is a spectrum of diseases, I think, some of which are in stage and nothing's going to work. If it's an infection, if it's asthma-related or inflammatory-related, then I think nitric oxide might be beneficial. We've talked about penile erection. There are a lot of other Viagra substitutes now coming on the market in the US and Europe. It participates in angiogenesis. If it participates in angiogenesis, it's probably going to play a role in wound healing. It'll probably play a role in tumor growth, solid tumors. We know it participates in the inflammatory process in atherogenesis. In the atherosclerotic plaque, there's evidence of excessive nitric oxide formation in superoxide and peroxynitrite because you see nitrated proteins. It inhibits smooth muscle proliferation, it enhances endothelial proliferation, it influences platelets, it inhibits their aggregation, and plays a role in polys and their tumbling. So remodeling of blood vessels and the atherosclerotic process, which is an inflammatory disease, there's a significant role of nitric oxide. And of course, other inflammatory diseases as well. If you could prevent the excess production of nitric oxide by NOS2 in these inflammatory diseases, maybe you can have some new drugs as anti-inflammatories rather than cyclooxygenase inhibitors or the COX inhibitors. It's very cytotoxic to tissues. It kills bugs, it kills tumors, it kills neuronal cells. So the key with nitric oxide is that if you have too little, you have disease. If you have too much, you have disease. So you want to be able to control tissues to make just the right amount at the right time. Otherwise, there's a problem. So there's some complexity in all this, obviously. So you need very specific drugs. The way you get specific drugs is by understanding all the biochemistry so that you can use those as targets to design your molecules. If you start using non-specific drugs, you're going to have a mess on your hands and a lot of side effects. Next slide. In tissue transplantation, there's evidence that there's an inflammatory component because of histocompatibility mismatches. You get cytokine release, you make NO, it's toxic to the tissues. Septic shock, when patients have severe infections, they induce NOS2, they make a lot of NO. The NO is there probably as a self-defense mechanism to kill the bugs and the infection, but you're making too much and the blood pressure drops, and these patients that go the intensive care unit into a septic shock have a mortality rate of about 70%. So if you can come along with selective nitric oxide scavengers or NOS2 inhibitors, you have a major contribution to the mortality in septic shock patients or patients with crushed injuries and auto accidents, etc. It inhibits platelet aggregation. It's an anticoagulant. This has utility in heart lung machines and in dialysis machines and things like that. Gene regulation is an evolving area. We know from my work that there are about 30 genes that are activated by NO and about 20 that are inhibited. This is going to be an important area for the future, perhaps. Stem cell application. We think that nitric oxide regulates stem cell growth and differentiation. That's, I think, going to be a very important area for the future with tissue replacement, gene delivery, etc. Well, thank you very, very much for your attention.
Presentation and lecture. And that unfortunately ends our audience today with 2 Nobel laureates. And I hope the guests here who have listened to the 2 lectures have learned something from our guest speaker. And thank you once again, Professor Brubenkin and Murad. And I hope the rest of your time in Khon, although it's very short, will be enjoyable. Now I would like to hand over to the Associate Dean of the Faculty of Medicine and also the Director of the Nitric Hospital, Associate Professor Suthi Kraitakul, who will formally thank our guests and also will present the tokens of appreciation to our speakers. Dr. Suthi Kraitakul, please.
Thank you, Associate Dean Dr. Nisini. Rarely is Khon University blessed with such renown and illustrious guest speaker. I speak to myself and I hope today's audience when they say that both Professor Brubenkin and Professor Murad have added our understanding of their field, laser and nitric oxide. Thank you both for taking the time come to our country and see us here in Khon University. Our thanks also to International Peace Foundation for arranging this lecture in the series Bridge Dialogue Toward a Culture of Peace. You designed to pursue world peace through greater understanding in the cause that life is the true spirit of the Nobel Prize and its founder Alfred Nobel's dream. Lastly, we wish our guest speaker the very best health and enjoy your time in Thailand. And please feel welcome to visit us in Khon in future. Thank you once again, Professor Brumbergen and Professor Murad.
May I invite Professor Brumbergen to receive our token of appreciation. Now I would like to invite Professor Murad also to receive our token of appreciation. Thank you very much for your attention, everybody. And also, actually from 15:30 to 16:30, we will have an informal discussion between